Welcome to tonight's event on molecular tools for wildlife genomic health and conservation. There's going to be 3 wonderful talks today, which will include one by Charley and Alex Cagan, and I'll be the first presenter of the night. My name is Patricia Brekke, and I'm a research fellow here at the Institute of Zoology, and I will talk to you today about the impact of genomics on reproductive health, mainly focusing on birds. Okay. So reproductive failure is ubiquitous across species. Every individual, species, and population experiences reproductive failure, including humans, where around 25% of embryos fail to develop full term. And that is despite having incredible amounts of investment in understanding fertility and reproductive failure. And that is because in mammals like us, early stages of development are really difficult to track and diagnose. So we miss a large proportion of the early reproductive failure that we experience. And that's why we focus, or our team focuses on birds, which are an incredible model system for understanding reproductive failure, because we can track these large yolky eggs that develop outside of the mother and can sample them from conception up until death. And also, birds are a great taxa to do this because we have a lot of life history information for the species, as it's one of the most studied groups globally. They also have highly conserved developmental pathways and genomes, which makes comparisons across species a lot easier. So birds are a wonderful model system to understand reproductive health and reproductive failure. And understanding reproductive failure varies across disciplines, or our need to understand reproductive failure varies across disciplines, from medicine to economics. But in conservation, it plays a crucial role because breeding success is the key to the recovery of endangered species. And it's the reason why some of the first conservation interventions were specifically tackling reproduction and breeding success. And this is an embryologist called Charles Otis Whitman, and he developed some of the first conservation interventions to try and prevent the extinction of the passenger pigeon, which declined in the early 1900s. So he tried to breed the last few passenger pigeons in captivity by making them produce more eggs by reclutching, and also cross-fostering the eggs to other species like the rock doves. And unfortunately, Charles Whitman was unsuccessful, and the last passenger pigeon, Martha was her name, died in captivity in 1914, and the species went extinct. And unfortunately, 100 years later, similar stories are playing across the world. So this is the critically endangered seaheck, or Guam kingfisher. It survived only in captivity and no populations in the wild for the last 40 years. And last year, for the first time in 40 years, they reintroduced seaheck to the wild. And for the first time in 40 years, it laid its first eggs in the wild as well, which was an incredible accomplishment for that conservation program. But unfortunately for seaheck, they have really high levels of hatching failure. Around 70% of the eggs that the seaheck lay fail to hatch. And in the wild, none of the eggs have hatched so far. So the issues that we faced 100 years ago to recover species are still the issues we're facing today. In our group, we did a huge comparative analysis across species of birds globally. And we looked at differences in captive and wild populations as well, and we found that hatching failure was directly associated with threat level. So that critically endangered species experience at least twice the level of hatching failure than least concerned species. And species in captivity experience exacerbated levels of hatching failure as well, because they don't experience the right breeding conditions. So there's a real need to develop conservation interventions that tackle hatching success in order to recover species from extinction threat. So one of the first questions that we ask to try and understand this relationship between species threat level and hatching failure rates is, why do eggs fail to hatch? We don't understand the mechanisms that lead to hatching failure. And that's because in the ornithological field, from the beginning, we have assumed that undeveloped eggs that look like the egg that you eat in the morning are actually infertile. And when we developed a technique to actually test this, we found that, in fact, most of the eggs that look infertile are actually early embryo death. So infertility is massively biased across most studies. And most of the reasons why eggs are failing to hatch is because of early embryo death and not infertility. And most conservation programs have been focusing on trying to improve fertility rather than trying to improve embryo development and survival. So we need to shift our focus as conservation practitioners from fertility to developing techniques that improve survival during the early stages of development. So early stages of development, or early stages of embryo development, are one of the most complex phases in an individual's life. And it's completely reliant on the genomic integrity of that individual for normal development to occur. So having high quality and large amounts of genomic resources to understand embryo development is crucial for us to understand why eggs are failing to hatch, particularly in threatened populations. But unfortunately, we have very few good quality genomic resources for wild animal populations, let alone for threatened species. And most of the genomic resources that we have are in wealthy nations, which are some of the least biodiverse areas on Earth. So there's this gap in data, and this bias in data is a kind of a block to enable us to develop conservation interventions, genomic conservation interventions that might help save species from extinction. However, there are a few species of conservation concern where genomic tools and genomic resources are currently available, and they've had an important role in rescuing some of these populations already in a diverse range of studies. So from understanding, for example, the role of sexually transmitted diseases in koalas like chlamydia, and sexually transmitted diseases and susceptibility to these diseases, to understanding the impact of mating systems on the transmission of facial tumor cancers in the Tasmanian devils, to artificial insemination, for example, in the New Zealand kakapo, and also trying to understand the repercussions of some of the conservation interventions that we expose species to, for example, genetic rescue and translocation. And in the Florida panther, genomic resources have helped us understand that genetic rescue was an appropriate tool for reducing some of the reproductive abnormalities that these species experience due to inbreeding. The hihi, which is the species that I specialize in, has also had some of these genomic resources in place. And that's because we have been able to, we've been able to develop these genomic resources because we have collaborated with the New Zealand government and also with local community and indigenous groups across New Zealand that have enabled us to produce long-term monitoring and sampling in these populations over the last 30 years. And that means that we have amassed an incredible biobank of tissue and blood samples, around 7,000 of them, that have enabled us to apply a number of molecular resources and develop a number of molecular resources for the species. And these have evolved over time from developing microsatellites in around 2006, 2007 that allowed us to build pedigrees for quantitative genetic analysis and look at the heredity of some of these reproductive traits, to single genes and some areas of the genome, to whole genome sequencing and SNP arrays, and more recently developing or assembling whole genomes for the species, reference genomes for the species for either sex, and developing chromosome, characterizing the sex chromosomes in the species and developing linkage maps, as well as developing other DNA-based biomarkers such as the microbiome data and telomere dynamics. And so they have been, these genomic resources have enabled us to look at where in the genome reproductive traits are associated with, and also to develop genomic inbreeding measures such as runs of homozygosity to understand the impact of inbreeding depression on reproduction. And initial information from the impact of genomic inbreeding has shown that inbreeding varies between the sexes and also at different stages of an individual's life, so that during embryo development, males are particularly vulnerable to inbreeding depression, while at the juvenile stage, females are more vulnerable to inbreeding depression. And we hope that because we've been able to extract DNA from very early embryos now, in the hihi, we will be able to apply these tools from conception across the whole of the embryonic development stages, all the embryonic development stages, and combine these molecular resources with the information we have of fertility, the variation in fertility and early embryo death in this population across time, and combine it with demographic information and social information to understand the mechanisms and drivers of infertility and early embryo death in wild populations of birds, to help us narrow down those conservation interventions to help reduce the high rates of hatching failure that the species experience, as hihi experienced around 3 times higher levels of hatching failure than outbred species. So thank you so much for listening, and thank you very much to the people who led this work with me and that I've talked to, all the work I've talked about today, and all our collaborators in New Zealand and the UK. And it's my absolute pleasure to introduce you to Charley Yen, who comes to us from Queen Mary University. She's a postdoc there working on turtles and epigenetics, and I'm really looking forward to your talk. Thank you, Charley.
Hi everyone, and thanks Patricia for the introduction, and today I'll be giving you a broad overview of how we use epigenetic tools for sea turtle conservation. So biodiversity is the amazing variety of life on Earth that sustains our planet. But as you all know, we're currently in the midst of a biodiversity crisis. For example, there's been a 73% decline in average population size just over the last 50 years for vertebrate populations. And this devastating rate of loss is largely driven by human activities, including anthropogenic climate change, which has received considerable attention as it pervasively alters the environmental conditions affecting all species, with ectothermic or cold-blooded species being especially susceptible. So when threatened by climate change, a population only really has 3 possible options, and these are to move to a more suitable habitat, to adapt in situ, or face extinction. Therefore, a challenging but essential goal of conservation is to predict which of these routes is most likely, to be able to help identify and prioritize the most vulnerable populations for conservation action. And the exploding field of conservation genetics that Patricia introduced has really been at the forefront of this movement, as genetic diversity is crucial to the adaptive potential and long-term resilience of populations. However, given the unprecedented pace and intensity of current environmental change, a lot of these classic genetic diversity-based metrics may fail to capture the early warning signals of rapidly declining populations that need immediate interventions to avert extinction. And this is where the emerging field of conservation epigenetics is well placed to help, specifically focusing today on DNA methylation, as it's the best characterized epigenetic modification in non-model species to date. So this is the addition of methyl tags to cytosine bases of the DNA, which influences gene expression regulation. So rather than affecting the DNA code letters, it instead regulates how the code is read. So as a simple example, if the promoter region of a gene becomes methylated, it will typically turn off expression of that gene. So DNA methylation offers a layer of molecular information that reflects the regulatory landscape of the genome. And this can be altered by intrinsic processes like developmental progression from embryos to adults, as well as environmental factors. So in humans, things like temperature, diet, smoking, and even social stress can induce changes in our own methylation profiles. So just like in biomedicine, DNA methylation offers useful biomarkers for monitoring wildlife, as it can reveal hidden clues about an individual's condition on a much shorter timescale than traditional genetic metrics. And it's also advantageous over looking at gene expression directly, as it's more stably maintained within the genome. So temporally, it can integrate information often across a lifetime, and spatially, there are usually useful cross-tissue correlations that can be leveraged. So minimally invasive tissues like blood can be sampled, yet still report widely on relevant traits. So we developed these concepts for sea turtles, which are a set of 7 endangered species facing many threats from human activities, including poaching, pollution, bycatch, and coastal development, to name a few. So as ectotherms, it's also important to understand the additional risk posed by climate change, especially as they have long generation times of up to 37 years, so this constrains their rate of evolutionary adaptation. And they're also some of the most loved and heavily managed wild species in the world, with extensive conservation programs running, as you can see here. So as we're super involved already, it's really important to make sure our current management strategies are actually appropriate for their biology. And our main study site is in Cabo Verde, which is just off the coast of West Africa and hosts one of the largest loggerhead nesting groups in the world. And this is where the Turtle Project was set up over a decade ago to work closely with the brilliant local NGOs to monitor and protect the loggerhead population. We have really successful programs already, such as anti-poaching patrols and hatcheries to mitigate light pollution and dog predation on nests. So now our attention turns to climate change, as it's still hard to incorporate into day-to-day conservation strategies due to some key monitoring challenges I'll outline next. So sea turtles are threatened by climate change at nesting beaches in 3 main ways, where the first is nest mortality, but as this is easy to see and quantify by eye, or easier to see and quantify by eye, a monitoring solution isn't really needed via molecular markers. Whereas the other 2 threats are cryptic, which means they're difficult or even impossible to measure via direct observation. And so these could really benefit from a monitoring solution, which we think could come from methylation markers. And we'll start with thermal stress, which is when embryos are incubated below the lethal thermal limits, but they can still suffer fitness costs due to developing under thermal stress, such as worse locomotory ability and body condition. And this is bad news for sea turtles, as soon as they hatch, they basically have to make a mad dash across the beach, all whilst avoiding their many predators, with a 1 in 1,000 chance of making it to adulthood. So any sublethal effects will be really detrimental to population recruitment rates and overall persistence. So to help monitor these cryptic effects, we set out to identify methylation biomarkers, which involve teaming up with the amazing local NGO Project Biodiversity on Sal Island. So this is where we relocated the clutches of wild nesting loggerheads to this in situ hatchery on a beach. We split the eggs randomly in half and then buried them at different depths to induce different incubation temperatures. And this is a powerful setup as it allowed us to manipulate temperature in field conditions whilst making sure insights are as relevant to real life as possible. And it also controls for the maternal and genetic effects through the split clutch design. Then upon hatching, we performed a series of fitness tests and collected a drop of blood from a subset of 40 for methylation sequencing. So this plot just shows that incubation temperature was indeed different between the depth treatments. And between them, we identified 287 differentially methylated sites where methylation variants could strongly discriminate the incubation environment experienced by the hatchlings. And these were distributed throughout the genome, including on genes with roles in neurodevelopment, musculoskeletal development, and cell growth and proliferation, as some examples. And so these functions support their role, these markers, as relevant markers of the sublethal effects we caught in our experiment, where, for example, the shallow incubated hatchlings in yellow were slower at self-righting than their sibling counterparts in the deep, cooler treatments in blue for each clutch. And again in yellow, the shallow incubated hatchlings were also slower at running overall, but with this interesting interaction where they got faster again towards the upper end of their temperature treatment range. And linking back to methylation, there are even some sites where there were direct correlations between methylation and fitness performance. For example, here on the TMEM273 gene, where methylation per hatchling interacted with runtime. And this really mirrors the interaction I showed earlier between runtime and incubation temperature. And so this is a cool result to us, as it suggests that we can find methylation marker sites that can directly report on the thermal stress effects of interest. So after briefly covering thermal stress, we can now move on to the third and final threat, which is sex ratio skew. So along with over 400 vertebrate species, sea turtles have a temperature-dependent sex determination system, or TSD for short. And this is where instead of having any sex chromosomes, their sex is completely determined by temperature during a thermosensitive period of egg incubation, with females produced at warmer nest temperatures and males at cooler temperatures. So, of course, with climate change, a lot of people are worried about the risk of extreme female bias leading to demographic collapse when there's no breeding males left. For example, there's a lot of studies similar to this one where they're projecting 100% female skew by the end of the century. However, it's unclear whether these projections accurately represent reality, as most of them have been modeled using nest temperature as a proxy for sex ratio with minimal empirical data, mostly extrapolated from a few constant temperature lab experiments performed many decades ago. And this is because there's still currently no method to determine the sex of a hatchling, a newly hatched hatchling, without sacrificing them, which is, of course, highly restricted as endangered species. With the only non-lethal method right now involving rearing the hatchlings for several months in specialized aquaculture facilities before performing this surgical procedure called gonadal laparoscopy. So this is also unfeasible at scale, especially for conservation programs. So with no genetic differences between the hatchlings, this is again where we think methylation can come to the rescue. And to identify markers, our collaborators at Florida Atlantic University incubated loggerhead eggs at 3 different temperatures. Then upon hatching, they collected blood samples from each of the hatchlings and then reared them for laparoscopy for sex verification. And this identified over 700 differentially methylated sites that could strongly discriminate between males and females, and so could act as our sex markers. And these were again distributed through the genome with diverse genes, including 4 with previous links to temperature sex determination in other reptile species, as well as other interesting candidates such as AMD-HD2, which was also found to be the strongest differentially methylated sites between children from both saliva and blood samples across ethnicities and ages. So this hints at its possible universality across tissues, populations, ages, and now even species. And finally, we applied our novel markers to sex hatchlings from a field experiment in Cabo Verde, which is really similar to the one I showed you earlier with shallow treatments in yellow and deep treatments in blue, just expanded across 3 time points over the warming nesting season to induce more thermal variation naturally. And compared to the methylation-based sex ratios, here with the y-axis showing the proportion of females, we found that traditional temperature proxy-based methods overestimated female production by an average of 52.4% and up to 77.8% for the exact same nests. So this is a striking conservation implication, as it suggests that current demographic projections really need recalibrating with empirical sex ratio data to recalibrate our management considerations. So to quickly summarize, I hope I've convinced you that methylation biomarkers offer a promising solution for monitoring the cryptic effects of thermal stress and sex ratio skew to ultimately advance conservation guidelines beyond mortality for sea turtles as climate change progresses. And for widespread deployment of our markers, we've been exploring some portable sequencing solutions, including Oxford Nanopore technologies. So this allowed us to successfully pilot sequencing on-site in Cabo Verde with just a single suitcase of equipment and go from sampling hatchlings on the beach to usable genomic data in under 3 days. With our long-term goal being to collaborate with our NGO partners to deploy this tool to inform hatchery management decisions in real time, such as nest shading design. And going beyond climate change, methylation markers can be developed for a lot more of a broad range of conservation relevant traits. So I've already covered sex and thermal stress, but they can also be used for other developmental states like age, reproductive state, or other external stressors such as pollutants, disease, and parasites, and other ecological history traits that are useful to conservation managers such as foraging history and population of origin. So the overarching goal of the Turtle project is to build a research program where you can essentially sequence any individual to get a methylation map of all of its conservation relevant traits to guide a precision conservation approach, just like in precision medicine, and ultimately improve conservation outcomes for sea turtles by catching and managing all of these early warning signals of silent threats to their persistence. And with that, I'd like to thank everyone involved in this project, and yep.
Thank you so much, Charlie. That was wonderful. And I'd like to introduce our last speaker of the day, Dr. Alex Cagan, who's here from the University of Cambridge, talking about somatic mutations in wildlife. Thank you so much, Alex.
Thank you very much, Patricia. I'd like to thank the organisers for the kind invitation to be here, and also thank you all for coming. It's a real pleasure to be here and talk about our research. And so my lab focuses on developing methods to study how aging and environmental factors impact the DNA in our cells as we age, and how this can impact the health of both humans and other species. And I kind of just wanted to start with this analogy of a palimpsest. I don't know if people are familiar with these. These are ancient documents, and in the past, these materials like vellum that were written on were very valuable and hard to get. So if you wanted to write something, you might just find an old piece of vellum, scrape off what was there, and replace it with the new text you wanted to write. But even when you did so, you would still have some of the traces left of the previous documents. So you can see on the left is an ancient Arabic manuscript, and then on the right, with some special imaging techniques, you can actually see there's still the trace of the previous document, which was a herbarium drawing of a medicinal herb. And if there's just one thing you take away from the talk today, it would essentially be that the DNA in our cells is essentially a living palimpsest. So the factors that, the different environmental factors, the process of aging, they all leave marks in the DNA within our cells. And so by looking at our own genomes and the different cells in our body, we can actually look back in time and learn about the different environmental factors that have shaped us as individuals and organisms. And that can teach us both about ourselves and about different environmental factors and how they're impacting our health. And so just to explain why I think this is the case, we have to go back to the beginning. So we all start life as a single cell, and we develop into these multi-trillion cell organisms with many different tissues, really multicellular miracles where all these different cell types are cooperating together, enabling us to exist. And this would all be well and good, except the DNA in our cells, which encodes this information telling our cells how to behave, it's constantly being damaged by a variety of different exogenous and endogenous forces. So as a result, we think about the 70,000 different lesions to our DNA in every single cell every day. And so you get this damage to the DNA. Fortunately, we have a variety of different repair pathways in our cells that are all designed to identify this huge amount of damage and correct it so that our DNA can go on functioning as normal. However, these different repair pathways act with varying levels of fidelity. So some of our repair pathways are excellent. They're very meticulous. They'll find that damage, they'll get rid of it, and you'll never know the damage was even there. But we have other pathways in our cells that are a bit sloppier. They're just concerned with getting the job done. The cell is a very active place. There's a lot of metabolism going on, so sometimes you just need to get rid of that damage off the DNA and continue. And sometimes that process leads to incorrect repair. So you get a C nucleotide being replaced with a T, you get a single base pair mutation. And through this process of ongoing damage and sloppy repair over time, we get mutations. It's this combination of damage and incorrect repair. And so as a result of this, as we age, all of us become these molecular mosaics. As you get mutations accumulating in our cells, those cells divide, and that typo in that original cell, that mutation, is then passed on to the daughter cells, and those cells spread throughout the body. Fortunately for us, we think most of these mutations have no impact on how our cells behave. Our cells can continue going on because not all of the DNA is functional. However, we know some of these mutations can change cellular behavior and cause some of our cells to start to go rogue and not cooperate with the rest of the cells in our body. And this is most famously known in the case of cancer. So some of these mutations can cause cells to keep proliferative signaling and dividing when they're not meant to induce local angiogenesis to get more resources. So the cells start to behave in a more selfish way, just as a result of this accumulation of mutations and some falling in regions of the genome that change their behavior. It's like typos messing up a cookbook and spoiling the recipe. And so, yeah, as I said, some of these mutations can lead healthy cells down this route of dividing, hyperplasia, and ultimately cancer. And so we've known about this for a long time, but what we'd really like to do is study what's going on in the genomes of these healthy cells before they transition to being cancer, and how do environmental factors shape this transition. But it's been technically very difficult to study these mutations because you can sequence a tumor and you can look for all the shared mutations that have arisen from the original cell. But when you're looking for mutations in healthy cells, you're looking for mutations that are just present in single or small numbers of cells. And just because of the accuracy of genome sequencing, that can be really challenging. And so during my postdoc, myself and others worked on developing methods that would enable us to look at what's going on in the genome of these healthy cells in normal organisms. And so to do this, we developed this pipeline where we can take tissue samples, we embed them in paraffin wax, and we essentially make microscope slides. This is kind of standard histology. And then the magic part really is that we use laser capture microscopy and some accurate genome sequencing methods to then isolate specific cell types or small numbers of cells within tissues and sequence them and count the number of and types of mutations that are accumulating. And just to show you a bit what this looks like, these are the microscope slides we make of kind of different tissue sections. And then the cool bit is that we put them under a microscope and we fire a laser beam through them, and we can isolate out small groups of cells. And just to show you that a bit more clearly, this is a video we took of isolating some spermatogonial cells in sperm from a seminiferous tubule. So this is the laser going around. It's fun to point with the laser at the laser. And So it cuts through the cells, and this slide is also on a plastic membrane. So the slide's upside down in the microscope, the laser cuts through the plastic and the tissue, and then if you're lucky, it falls off, but there's a lot of static sometimes, and these are very tiny, thousandth of a millimeter. So we blast it with a wider laser, and that's called laser catapulting. And the idea is if you're lucky, it'll fall into your collection well, but half of the time when I was doing my postdoc, there would be a little air current, and they're basically like little sails, and they would just fly away, and we never saw them again. So I spent a lot of time just hoping that there was like a little fleck of dust in the wells. And if there was, we'd lyse the cells, isolate the DNA, sequence them, and then we could start counting and looking at what are the mutations that are occurring in these cells, in different tissues in the body. And so myself and others, we did this across the human body because there was nothing really known about these processes in healthy cells. Different people taking different cell types and tissues, looking at individuals of different age and kind of charting these, we call them somatic mutations because they're within the soma or the body, and looking at what happens to these cells in terms of the mutations with age. And just to kind of, before we move on to the cool animal stuff, just the highlights of what we found in humans is that mutation rates differ significantly between cell types. So we have very rapidly dividing cells in our body. For example, in our intestines, the epithelial layer divide about once every few days or once a week. And each of these cells, the stem cells in our intestine, get about 50 mutations in the genome every year. But then if we look at cells like in the blood, we get about 20 mutations. And interestingly, even non-dividing cells like the neurons are getting about 17 mutations every year. So this ongoing damage and repair doesn't require cell division. It's happening in every cell. And kind of leading back to Patricia's talk earlier, there's much lower rates of mutation in the germ cells like the sperm and the oocytes. And this makes sense because these are really the immortal cells that are passed on to the next generation. And mutations can have profound consequences for fertility and health of offspring. So it seems like our bodies invest a lot more in protecting these cells. And unfortunately for us, our soma is a bit more disposable. There's less investment in protecting our soma. But as well, this is kind of the key for how we can use this for environmental monitoring, is as well as counting the number of mutations, the types, different types of environmental exposures can produce different types of mutation, and we can study that by sequencing the genome and infer the types of processes that cause those mutations in the first place, so a bit of kind of genomic archaeology. And this works because you can see, for example, here, if you get a C to a T mutation, every time there's a mutation, we can look at the letter before and after it, and we call that the trinucleotide or three-letter context. So here we have a C to T mutation that's occurred with a T to the left of it and a G to the right. And different environmental factors will tend to cause mutations in different three-letter contexts. So for example, UV radiation, due to the kind of biochemical processes that this kind of damage causes, will tend to cause C to T mutations. And this is the kind of the different locations they could occur at. They'll tend to be at these specific trinucleotide contexts. Whereas if you have exposure to something like cigarette smoke, it'll tend to cause C to A mutations at a very different context. And we can look at this mutational spectra, and then we can infer what was the actual process that caused the mutations we're observing. So if you were to sequence skin cells, you'll see a lot of these kinds of mutations caused by UV exposure. Whereas if you sequence the lungs of a smoker, most of the mutations you'll see will be this signature related to cigarette smoke. And so this gives us a method, in a way, to look at a genome and learn about the environmental history of the cell that we're looking at. And kind of linking to health and conservation, why is this important? Well, we know that environmental pollutants are a growing health concern. There's estimated to be about 350,000 different synthetic chemicals that we've created and used globally in our environment. And for most of these, unfortunately, we're not very clear on what their environmental and health consequences are. And one way we can learn about this is to sequence cells that have been exposed to some of these chemicals and look and see, are they mutagenic? Are they producing these specific types of damage that we can identify? And so there are different groups who are creating catalogs of these different types of exposures, organic industrial pollutants, different types of radiation or heavy metals, and many of them do have these kind of specific, unique spectra or signatures. And so when we sequence genomes, we now have a catalog we can look to. So we can look at human cells or cells of other organisms and see, are we detecting any of these? We can use that as a kind of genomic monitoring. So in a sense, every cell in the body becomes a potential biosensor when we look at the genome. And kind of touching on this, there's a big mystery in the field of environmental pollutants, which is actually, when we look at different carcinogens that when you're exposed to them can increase your risk of cancer, the kind of easy understanding we have, what makes sense is when they're mutagenic, because we know the more mutations we have, the higher the risk one of those mutations will cause cancer. But actually, it seems like most carcinogens are not causing any mutations. So the question becomes, how are these different environmental pollutants that aren't causing mutations increasing our risk of cancer? And to understand this, we have to understand another aspect of what goes on in our cells as we age, And that's that once mutations occur, as I kind of alluded to earlier, some of them will cause cells to change their behavior and clonally expand, so that as we age, we get these patchworks of clones. But whether or not a mutation causes a cell to expand or not is not always a cell intrinsic property. It can be shaped by the environment that cell is in. Whether there's a lot of inflammation or other factors can determine whether a mutation is going to just sit there or whether it's going to trigger that cell to expand. And so, as we age, this is an illustration, but it's reflecting real data of these kind of clonal expansions we get in our skin tissue as we age from a study we did in the eyelid. So they're invisible to us because we can't see our genomes, but all of us as we age get these clonal expansions of cells that have carcinogenic mutations. And we think that we're learning how different environmental factors can accelerate or decelerate this process. And one really nice recent example of this was looking at the effects of urban air pollution. So just to talk a little bit about this, one of the key features of urban air pollution are these very tiny particles called 2.5 PM based on their size. So much smaller than a human hair, even smaller than a red blood cell. And we know that ingesting these can cause a variety of different short-term and long-term health consequences. And one of these consequences is increasing the rate of cancer. But it seems that these particles are not mutagenic. So when we expose cells to them, we don't see an increase in mutation rates. And a group at the Crick in London did a lot of beautiful work, and they figured out what's going on in this case is that in our lungs, we already have cells, a certain population of alveolar cells that are normally involved in wound healing. But if they already have a pre-existing mutation in a particular gene, then if you ingest this particulate matter, this air pollution, these cells will start to expand. And the reason they do that is because when we breathe in this particulate matter, our immune system thinks that it's some kind of invading virus, and the macrophages start to attack this particulate matter. When they do so, they'll release these cytokines and interleukins that will trigger localized inflammation because they think they've got to protect you from this particulate matter. But these interleukins will interact with these cells with this mutation, and if the cells have this mutation, they will, in that interleukin environment, start to clonally expand and spread, and ultimately that can lead to cancer. So it's giving us a much more nuanced understanding of how different environmental factors can impact our health. You can have environmental pollutants that just give you more mutations, and that can increase your risk of cancer. But it seems that a far larger number of environmental pollutants aren't actually causing mutations, but they're impacting our tissue microenvironments, maybe through causing chronic inflammation or other factors like this, and that can really shape the trajectories of our cells. And so really, this approach of now being able to study somatic mutations within our cells as we age, I think is giving a slightly different perspective on genetic variation. So when I was learning genetics at school, we had this idea that we all have one genome inherited from our parents, and that's the genome our bodies carry throughout life. But now we know it's a bit more nuanced than that. When you look a bit closer, actually, we think no two cells in our body share the exact same genome because of this accumulation of mutations over time. And we're learning how different environmental factors can shape this. And so far, almost all the work in this has been in humans. But there's very good reasons to look beyond that at other species. And what can we learn from other species? One of the things we've been very interested in that I'll just go over very briefly is lifespan. So we think these mutations can impact our health as we age. And when we look across species, we see this enormous variation in lifespan. So we wonder, are species with longer lifespans much better at protecting their DNA than species with shorter lifespans? We did a really in-depth study with collaborators here at London Zoo to address that, where we looked at cells from very short-lived and very long-lived species to try and see if there were differences in the way they protect their DNA. We looked at 16 different species in total, and just the take-home message from that study was that we actually found for the first time that species with shorter lifespans are accumulating mutations at a much faster rate. So just to summarize this data, humans in our intestinal cells get about 40 mutations every year, but then as species get shorter lifespans, that rate increases, and mice get about 800 mutations in their cells every year. So it does seem that short-lived species invest less in protecting their DNA from damage, probably because they don't need to, because they're not going to be around that long. But this finding opens up obvious potential applications of this fact. So the fact that shorter-lived species accumulate mutations at a much faster rate means we could potentially use them as much more sensitive monitors of how different pollutants are affecting our genomes. And so this comes into the topic of sentinel species. So you're probably all familiar with the canary in the coal mine. It's the classic sentinel species, and it works because if you're going into a mine where there's a CO leak or a carbon methane leak, you're going to detect it much faster with a species with a higher metabolism. So the canary has a much higher metabolism, so this is a gas leak, it's going to pass out much faster than the human. And so these sentinel species that are much more sensitive than we are to environmental perturbations can really be informative to us about changes in the environment and how they're impacting our health. And so now that we can measure the genomic changes in the cell of any tissue in any species, it now opens up this opportunity to use different cells and somatic mutations from different species to learn about these impacts in the environment. And just kind of to very briefly wrap up, this is very new technology, so we're just starting to explore this in other species and what we can learn from this. But we're doing a collaboration with Patricia Brekke to look at potential impacts of air pollution on brain health and how we could use different sentinel species and genomics to investigate this. So I touched on air pollution earlier in the context of lung cancer, but it also has far other more pernicious effects, including on brain health. There's many different ways that it's been studied that it can increase premature deaths, reduce cognition, and increase rates of disease conditions such as Alzheimer's or Parkinson's. And here's a map on the right of different emission levels from around different parts of the UK, and you can see real concentrations in urban areas. And so to study this, we think an excellent sentinel species would be the grey squirrel. And why do we think the grey squirrel would be an excellent sentinel species for this? Well, they're very geographically widespread, so they're found across the UK. They have relatively small ranges, which makes them superior to humans, because the challenge with humans is we move around throughout our lives. So it's really hard to correlate these exposures to know exactly who's been exposed to what. But these squirrels have very small ranges. They spend their whole lives in a small area. They tend not to disperse very far. They're invasive, so their populations are controlled, so we can get access to tissues. And they're also rodents, so we can compare data in squirrels from laboratory animals like mice. And so one of the studies we're embarking on to try and look at how we could use this genomic information to monitor both human and animal health is to look at the brains of these squirrels. And one of the approaches we're taking is, so I talked earlier about this laser capture microdissection. So we can take sections of the brain and we can isolate specific areas. But what's really cool that we can do with the squirrel brain is we can also do metallomics. So we can take different slices of the same brain and we can look at the mutations in one, but we can also use a very different approach to very precisely quantify the concentrations of different metals that we can measure in different regions of the brain. So this particulate matter, which is often different small amounts of metal, will deposit in different regions of the brain at different concentrations. And so we can take squirrels from urban and rural areas, very precisely measure the levels of different metals that have been laid down, and then from an adjacent section, we can isolate tissues from high and low metal concentration regions to very precisely measure within those squirrels from different areas, is there a mutagenic effect or some kind of other effect on the genome of these heavy metals? And we can apply this in the squirrels as a case study, but we could also apply this potentially to many other species. And so in summary, I think this is really just hopefully the tip of the iceberg, that now we have these new genomic tools and other tools like epigenetics, we can start to really look in detail. And almost any species could potentially be a sentinel. We could look at species for aquatic health, for air pollution. We could look at lichens. You can also look at how other environmental factors like stress may be impacting these processes. So I hope this is really just the tip of the iceberg, and that in the future we'll be able to come back and learn far more about how environmental factors are impacting our health through these new genomic methods. And so with that, I'd like to thank all the people who've made this possible, including many key collaborators at ZSL. And I look forward to a panel discussion, and thank you very much for your time.
Alex, that was wonderful. And shall we go and sit down on the stage and then we can maybe have some questions?
You mentioned using gray squirrels to study brain impacts from air pollution and other environmental factors. Are there any studies or recent research that have been done, and what were the results of that? Or is it just in the modeling phase right now?
For the grey squirrel, this is, for the genomic side, this is just in the kind of initial phases right now. Okay. The samples come from Patricia's lab, so I don't see anything you want to add about measurements that have been done in the grey squirrel.
Yeah, so we currently have a study looking at mapping all 20 different metals across all brain regions of the grey squirrel in populations at a gradient of urbanization and air pollution from the centre of London all the way out to some of the most rural areas in England and Wales, so North Wales and the north areas of England. And we see a direct correlation between some of the exogenous metals, heavy metals in the brain, and proximity to main roads, so vehicle emissions. Pollutants are the main ones that we're finding accumulating from the olfactory bulb, which is at the front of your brain, to the cortex at different concentrations.
And just to follow up, thank you, but so have you are there any models to then study kind of like the behavioral effects of that heavy metals, or is it just more so focused right now on like, yeah?
At the moment, we're just developing the baselines for the species, but yeah, of course, at some point in the future, we'd like to see whether we can test the cognitive effects of exposure to metal air pollutants on behavior. Yeah.
All right, thank you so much.
I'm just wondering whether you could give some examples of specific conservation interventions once you have all this incredible information from the eggs of the hihi and so forth. I've been to Tiri Matangi, by the way, the island, amazing place. And unfortunately, I haven't been to too many sea turtle nesting sites, but maybe you could give us an example, or at least allude to what you might want to do with the information. So it could go for any of you, I guess.
Do you want to start?
Okay, yeah. So it's pretty early days in terms of this kind of monitoring, but there have been some examples, not so much from a conservation angle, but from a health angle. For example, with cattle. So this method was used to kind of pinpoint. There were some populations of cattle that were having very high levels of intestinal adenocarcinoma and stomach cancers, and it wasn't certain what the cause was. And through this genomic sequencing, it was pinpointed to this black fern that was in the area where some of the cattle were kept that they were eating. And so they noticed this unusual mutational signature in the stomach, and then they were able to pinpoint an extract from this black fern. When you expose cells to this black fern extract, it caused the exact same spectra. And so that's kind of been used to change some of the management practices. But that's a kind of specific agriculture example, but that's kind of the goal here, is that by monitoring, you can start to alert yourself to the causes of some of these different pollutants, pinpoint what they are, and then use that to change management or kind of environmental practices to try and minimize those pollutant exposures.
And for birds, in terms of early developmental pathways, the interesting aspect of that is comparing across species, so understanding kind of the genomic mechanisms that drive those changes in early hatching failure. So it could be that those developmental pathways are broken somehow, or the integrity of the genomic pathways are broken somehow once populations decline because of high levels of inbreeding interacting with genetic diversity at those kind of early stages of development, the impact of inbreeding on genetic diversity, so that those regulatory pathways are not working properly. So trying to get to the mechanisms that are driving kind of that mortality is kind of what we're trying to achieve.
And yeah, for mine, I started to allude to it in the presentation, but it's nice to expand. So our main next focus will be hatchery management, because, as I mentioned, so these hatcheries are all across the world. So they've mainly been developed for things like light pollution from hotels and coastal development, but there's been no integration of how climate change is impacting the environment that these nests are being incubated in. So they're often at the back of a nesting beach, which is really hot and dry compared to their natural environment. But people are seeing high success rates, so they're like, yeah, great, we're doing great. But we want to add this monitoring aspect to check if the sex ratios are okay, if the hatchlings are thermally stressed, and hopefully be able to guide, for example, nest shading design, and some people even try and cool the nests with water. We want to see the effects of that. But yeah, currently there's a lot of conservation programs that are just running blind based on what they think is good, but no one actually knows what's going on behind the scenes.
Some really good talks there, thank you. I have a question for Alex about the methods that you're using. Why are you doing the laser dissection rather than something like single-cell sequencing?
Yeah, it's a great question. We would love to do single-cell sequencing if we could, and that early study was before even further improvements in methods. So sometimes we can do single cell now, but the issue was that it still wasn't accurate enough to just rely on the mutations in a single cell. So we would actually isolate groups of cells that were clonally related. So like intestinal crypts, you have a basal stem cell that divides a lot. So if you cut out one crypt, all of those cells will share the mutations that were accumulating in that stem cell. So it's a kind of way of cheating and getting multiple copies of essentially a single cell. So it's just a technical issue to overcome this challenge of sequencing is not as accurate as we'd like it to be. But the fact that it's getting more and more accurate every year, and all these amazing technologies like being able to use it in the field, which is something we only used to dream about, is really opening the doors for the kind of things we can do.
Thank you.
Hi, thank you for the presentation. I'd like to check my understanding first. So for the methylation, basically, if you want to make make sure that this methylation on this side is directly you have to prove it's causational to like the environmental factor, do you like need a very large body of data and study for that? Like, or is it like you can?
So yeah, it's a good question. So yes, if you want to prove causation, you're going to need very controlled lab experiments, necessary fission, that kind of experimental design. For us, we're mainly looking for correlative biomarkers that can improve conservation. So we know that we can't prove like if it was the cause or the effect, but for our purposes, as long as it reliably correlates, we're happy to use it. And then in terms of sea turtles, there are other study systems like freshwater turtles where you can run these really cool controlled experiments, and they are proving methylation is causally added in response to thermal stress, for example. So, yeah.
Wow, that's very interesting.
Yeah,
Thank
so you can try and draw parallels, but yeah, we can't. Sea turtles are really protected, so this is about the limit that we're allowed to do.
Yeah. And also building on that, like, can you use like data from other species? Like, are they how much can you reference between different species?
Yeah, so you can quite generally relate, I guess, mechanisms of epigenetics within, I would say, probably reptiles, because methylation is very taxa specific. There's a lot of papers doing comparative epigenomics across the animal tree, and plants are a whole nother kettle of fish, but kettle of plants. So yeah, you want to be quite specific, but the mechanisms of like methylation, for example, within vertebrates is fairly well conserved, but to get the specific levels of methylation and exact regions that are responding, that would be species specific and even population specific, because it's shaped by local adaptation, the methylation platforms themselves.
Thank you.
Thank you for this talk. This is like my first one here. In my heart, I really care about the climate change. Okay, so I'm talking to all of you and all of us in this room. If we're doing all these kind of examinations, tests, etc., how are we presenting it to the bigger world to make it better for the world to come, such as the blue tortoise? We want to be able to see them reproducing, them having more. Any species, we want more of that species because, look at the dinosaurs, they wiped out. So that's one thing, is how we're going to be helping these animals, mammals, to have more, which is good. The second is like we're doing this test examination on the rats to find that it's very similar to human beings. That's helpful. And then the same is to like see why the eggs are not hatching, which is useful as well. But how are we using all this information to go to the government to stop hurting our world, to stop hurting our animals or the things that we really need in the world, to make it beneficial? How are you, or how are we? Like, we need to see it done. We are in a climate change, and we need action. Thank you.
I guess from two perspectives. So first is by improving the kind of conservation practices that we have so that we can produce kind of healthier, better populations of animals that are threatened with extinction. That's kind of, I think, one of the main targets of all of our research. And I guess the second is that we do try to communicate our science to policymakers so that they improve health for everyone, not just for humans, but also for wildlife. And so, for example, some of the work that we're doing on pollution, having evidence of the impact of air pollutants on brain health in squirrels actually has quite a lot of repercussions for human health, but we can't do the level of detailed studies on humans that we do on wild squirrels. And they're exposed to the same complex pollutants that we are. And so that provides another line of evidence for policymakers to change policy around air pollution exposure for humans and for wildlife. So I think that's kind of the from my perspective, the kind of two main areas that I think we're focusing on as scientists.
Yeah.
Yeah, I don't know if you guys want to add.
Yeah, I mean, yeah, very well put. I can fit my thoughts into those two angles too, which the first is, yeah, there's limited time and resources to avert this biodiversity crisis and all of the threats facing them. And we are spending the money, so we might as well be finding out how we best help them the most efficiently and the most appropriately, especially, as I say, for sea turtles that are so loved, that people are doing so much for them already. We got to make sure it's correct. And yeah, like a specific example from my work is the sex ratio skew. So everyone thinks that they're all going to be 100% female by the end of the century. Everyone's kind of panicking, spending loads of money, moving the nests, doing a whole lot. But that turtles have been around for over 300 million years. They have persisted through massive climatic shifts. So they probably do have adaptive mechanisms we just don't understand yet. So we need to understand that to understand, do we actually need to do anything? And if we do, what do we do that we're not actually hindering their adaptive processes, for example? And then the second point, so in Cabo Verde, we work closely with, I showed all the NGOs, there's so many on the islands, and so we try to integrate with them as closely as possible. And yeah, our research helps them get funding, and then they can have more money to implement the conservation recommendations that we also find helps the turtles better. And with that, we also work with the Cabo Verdean government as well. So, yeah.
Yeah, I think you guys both said it so eloquently, so I don't have much to add, but I think it's just key to also just to have that data to actually measure things accurately so that we can make a stronger case. And from the environmental pollutant side, most of the studies so far have focused on, you have a mouse in a cage in the lab and you expose it to one pollutant at certain doses, but it doesn't really capture the complexities of real life exposures and how those are being affected by temperature changes. We really live in a super chemical cocktail. We're not just exposed to one chemical. We have long lifetime, low chronic exposure to multiple. And so we really need to get data from the wild situations and from urban areas and different places around the planet and actually understand what's going on in wild populations, because it's not really fully been captured by laboratory conditions.
Hello, thank you for the excellent presentations. I'm going to ask a question which was asked of me, and I didn't have an answer to this. So we release tens of thousands of turtle hatchlings. Now, unlike with birds, they're not. With birds, you ring captive bred or wild bred fledglings, and you can monitor them and see whether they survive to adulthood and whether they breed. Mm-hmm. So with the millions of turtle hatchlings that have been released, the hope is that they reach adulthood and they come back to their natal beach. But given that we're not marking them, is there any evidence that even one of those hatchlings ever reached maturity and whether they ever came back to their original nesting beach? Is there a way to identify them now?
Yeah, that's a great question, and you've touched on one of the main mysteries in sea turtle research, which is, as soon as you release those hatchlings, they enter what's called the lost years, where no one knows what they're doing until about 37 years later when they're mature enough to go back to breed. And so we can learn a bit of information from their genetics. So like we see, for example, very genetically distinct nesting groups. So they are coming back to breed, I guess, it's called philopatry. They return to their exact nesting beach to breed. So we know, they're coming back, and we can track based on the ocean currents and where the reproductive adults are coming from, that they're likely in Cabo Verde coming from Senegal, but in other places, there's a feeding ground and a nesting ground. But in those lost years, no one knows what's happening, and that is really difficult. And that's why we push for not just looking at mortality for these conservation measures, because, again, maybe we're sending out loads of really unfit and only female hatchlings, and then who knows what's going to happen in 37 years' time.
Yeah, so my question was not so much the lost years,
okay,
but do we know whether the ones that come back, is there even one from a turtle hatchery release that came back, or the wild ones who were never released from a hatchery?
It's just too long of a time span that, yeah, there's no exact turtle, and you can't really mark them for life very well anyway. I'm sure some people have tried and are waiting decades. But yeah, not yet, no. We've tracked them for a couple weeks with like nano tags to see where they go, and they follow the ocean currents basically, but beyond that, no one's tracked.
Well, if there's no further questions, I'd like to thank Charlie and Alex for their amazing talks and for coming today. And thank you all for coming. Really enjoyed having you here. Thank you.